So thank you very much, Adriano. Adriano, thank you very much for the presentation. And I would like also to thank the organizers for giving me this opportunity to share with you my results. As Adriano has mentioned, I will be discussing how to model chimeric antigen receptors and T-cell bispecific therapies in mice. As Adriano has mentioned, I work at the Valdebrón Institute of Oncology, but 1 year ago, I also joined the Hospital del Mar Medical Research Institute as research director. This is a map of Barcelona, and as you can see, my lab now is shared between two institutions, one on the mountainside and one on the seashore. So just to focus your attention on the models that we are going to be discussing, as you know, many tumors, approximately 3% of all tumors, are characterized by the amplification of HER2. HER2 is the driver, this tyrosine kinase receptor is the driver of these HER2 positive tumors. The most famous ones are breast cancers. Approximately 15% of breast cancers are driven by HER2, or gastric cancers. We and others showed many years ago that in addition to the full-length receptor, these tumors, some of these tumors expressed a truncated form of HER2 that is known as P95 HER2. And as shown in this slide, this is a very potent oncogene because this fragment is endowed with the ability to form disulfide bonded dimers. So it's a very potent oncogene that is co-expressed with HER2 in some HER2 positive tumors. We reasoned already several years ago that because of the truncation in this extracellular domain of P95 HER2, some epitopes exposed in the fragment would be masked in the context, in the 3D context of the full-length tyrosine kinase receptor HER2. And that turned out to be true. We developed antibodies against the peptide that is mapped in this slide. And it turned out to be that those antibodies bound the fragment, but did not bind the full-length receptor. So in other words, we had a specific antibodies against the truncated form of HER2. So a couple of years ago, we and some collaborators showed that in contrast to HER2, which is expressed in normal tissue, it is overexpressed in tumors, of course, but still normal epithelial tissues express detectable levels of full-length HER2. In contrast, there's no P95 HER2 whatsoever in normal tissues. We showed here by Western blot, also by immunohistochemistry, as you can see, with anti-HER2 antibodies, we do see positive signal, the brown signal in normal tissues, but with anti-P95 antibodies, we don't see any specific signal by immunohistochemistry or Western blot. So basically, we think that this is a bona fide tumor-specific antigen that is not expressed, at least at detectable levels, in healthy epithelial tissues. We think that this is important because, as you may know, one of the main hurdles in the development of chimeric antigen receptor or T-cell bispecific antibodies to treat solid tumors is that there are very, very few specific antigens of tumors, truly tumor-specific antigens. And that's why most of the CARs or TCBs are generated against tumor-associated antigens like HER2 that are overexpressed in tumors but still expressed in normal tissue. And as is highlighted here in several quotes from recent reviews, this leads to frequent fatal toxicities, severe adverse events, on target of tissue lethal side effects, anti-immune toxic effects. So we thought that the description or the identification of P95HER2 was a very nice way of targeting specific tumors. So in collaboration with Roche, we developed bispecific antibodies, and here the structure of those bispecific antibodies is shown. Using the sequence of the P95HER2 antibodies, we constructed these 3 specific antibodies that bind with 2 arms to the tumor-specific antigen, in this case P95HER2, and with 1 arm to the CD3 subunit of the T-cell receptor, so they can recruit T cells independently of the specificity of these T cells to cells expressing tumor-specific antigens, in this case P95HER2. So we had of course the challenge once we made these antibodies on how to characterize their activity, and that is when we became interested in different models. Of course, the in vitro models are quite straightforward. You just take your target cell expressing, in this case, P95HER2, and you co-culture them with lymphocytes from a healthy donor, and with or without your bispecific antibodies, and analyze the killing of these cells. In vivo, it's a little bit more difficult. This is taken from a recent review on the different animal models that can be used, from very simple models in which NSG or other strains of immunodeficient mice are injected with PBMCs from healthy tumors, from healthy volunteers, with your cells, until more sophisticated ways like like mice humanized with CD34 positive hematopoietic stem cells. So we started with the simplest model to analyze this bispecific and the activity of these bispecific antibodies. And this is just, as I have just mentioned, to inject human PBMCs into immunodeficient mice, in which we have implanted cells expressing our tumor-specific antigen, in this case P95. As you may know, in these mice, and this is showing in the white bars the percentage in normal blood, in normal human blood, and in yellow the percentage after 3 weeks of injection in the mice. Sorry, the legend is hidden. And as you can see, most of the human hematopoietic cells circulating after 3 weeks are T cells. So this is a very simple model, but it works very well to try and see if the T cells are recruited to the tumor. The only problem that we found, or the only issue that we had to address, is that in this case we used MCF7, a cell line overexpressing P95HER2. And as you can see, depending on the donor of the PBMCs, we saw some kind of rejection of the tumor, that it was due probably to mismatch in the HLA of this specific cell line and the specific donor. So it's a good idea to use different donors, because the degree of mismatch and therefore of rejection of the tumor may be different. But in any case, using here 2 donors, one of them that did not reject the tumor and one of them that had some degree of rejection, as you can see when we treated in the red curves with the P95HER2 TCB, we were able to see a very nice anti-tumor reaction. And I will not go into the details, but we showed that the lymphocytes were correctly delivered to the tumors, and there they could kill cancer cells. Using the more sophisticated humanization with CD34 positive cells, here again we have the comparison of the white bars, that is the composition of those cell types in normal human blood, with the composition that we see in mice 20 weeks after injection or 40 weeks after injection. As you know, in this model, lymphocytes get autoimmune lymphocytes get restricted, so we have at the end of quite a stable model in which we can perform long-term experiments. At 20 weeks, we see predominantly B cells, as you can see in the orange bars. At 40 weeks, in the red bars, we see predominantly T cells, some NK cells, and some monocytes, and very, very few granulocytes, if any. With this model, we tested the efficacy of these T cell bispecific antibodies against brain metastases. As you may know, this is one of the unmet clinical needs in breast cancer patients. Most of the antibodies do not cross the blood-brain barrier, and it's not easy to treat these metastases. And we were able to show by implanting cells labeled with the surface that the TCBs could guide the lymphocytes through the blood-brain barrier, very likely because they induce some degree of activation that allow the lymphocyte to cross the blood-brain barrier. In any case, they were quite effective against brain metastases. Taking one step further, these models, we wanted, instead of using cell lines overexpressing the antigen, to use patient-derived tumor xenografts. As you know, these are pieces of tumors that are implanted into immunodeficient mice, and they can be perpetuated. And we have a collection of patient-derived tumor xenografts positive for HER2, as shown here. Some of them are positive for P95 HER2, some of them are not. And this is a much more relevant model, much closer to the real tumors than cell lines that are quite homogeneous. Here we see quite a lot of heterogeneity reflecting exactly the situation in real tumors. Using these models instead of cell lines, we were able to show that the antibodies were also effective, and we also saw something that we cannot fully explain, is that many of the PDTXs did not grow into fully humanized mice. So the engraftment was very inefficient. Many of the tumors didn't grow, and sometimes we had to inject dozens of mice to have some tumors grown. We are still puzzled by this effect that we didn't expect. Somehow the tumors were rejected or were not allowed to grow as many of the patient-derived xenografts into humanized mice. With the ones that we could test, as shown here, we could show that the TCB was effective, and we could analyze the infiltration induced by the TCB in the tumors. So what we did was to systematically analyze the efficacy of the TCB in PDXs. We used again the PBMC model, and we were able to show or to determine the threshold of expression of P95 in order for the TCB to have anti-tumor effect, as shown here with a PDX that is positive for P95 HER2 and a PDX that is negative for P95 HER2. So basically, this is a summary of the development of these TCBs. And very recently, we wanted to expand this field of research by generating, in addition to TCBs, CARs against P95 HER2. And also we are very interested in modeling these CARs in our preclinical, in our mouse models. So basically the idea is similar. We took a bunch of P95 positive antibodies. Sorry about this. Yeah, there's a slide that is missing. That's okay. We had several P95 positive antibodies, and we made several CAR-Ts out of them. Basically, we sequenced these anti-P95 antibodies and generated, as shown in this slide, different second-generation CAR-Ts. Basically, the antigen binding domain of the antibody is fused to the transmembrane domain of the T-cell receptor and to signaling domains from the T-cell receptor and some coactivators, either CD28 or 4-1BB. And every time a lymphocyte that expressed this chimeric antigen receptor finds the antigen, the lymphocyte is activated and kills the target cells. So as I said, we took a bunch of anti-P95 antibodies, generated several CARs, and also humanized the sequence of these CARs. So we basically used the human sequence for all the non-variable regions and only kept the variable regions to minimize the development of anti-mouse antibodies when we use this in the clinic. Comparing several of these CARs, again, in a simple model, just we implant the cells expressing P95, and then we inject in these mice carrying the tumor the T cells transfected with the CARs. And as you can see here, comparing two CARs that were selected for in vitro studies, one of them, the 14D, turned out to be much more efficacious than the 15C. This is the way in which we analyze the efficacy of CAR-Ts. And this is the infiltration of CD3 positive cells in the right hand in the tumors. So again, another comparison, if we take now 2 degrees of humanization, you can humanize antibodies at different degrees, closer every step to the human version. And as you can see here, one of the versions, the H114D, H114D, sorry, was much more efficacious than the H214D, again, allowing us to select this CAR that we hope to test in the future in clinical trials. So at the beginning, we thought that with this model, we were going to have the same graft versus host that characterizes the PBMC model. I have not mentioned it, but I mean, the experimental window that you have in this PBMC, as you probably know, is more or less 1 month at most. After that, there's a massive graft versus host induced by the PBMCs. With CAR-Ts, this does not seem to be a problem. The number of T cells is kept somehow very low, and it does not cause graft versus host. And as you can see, we are very happy to see that even after almost 1 year, the mice are alive, and in this case, are free of the tumors induced by these cells expressing P95 HER2. Again, as I said, there are cells, CD3 human cells, after these many months, but they are in a low enough number that it doesn't cause graft versus host. Following up this long period, we were able to see some tumors that escaped. And of course, we were very interested in knowing mechanism of resistance. In this case, the mechanism of resistance was extremely straightforward. They just had lost the antigen, P95 HER2, as shown in these bars with the control cell, the cells that we used for implantation. And as you can see, the cells that we recover after the development of this resistance had lost the antigen. Anyway, again, we went from cell lines, as I have shown with the TCB, to PDTXs, patient-derived tumor xenografts, again, much more heterogeneous. And as you can see here immediately, unfortunately and as expected, the effect on PDTXs is much more slight, is not as efficacious as the effect on the homogeneous cell lines that we make. This is somehow makes sense in the sense that as you know, so far, CARs, chimeric antigen receptor, have not been efficacious in any clinical trial. There's not, on solid tumors, sorry, there's not a single CAR T approved for solid tumors. They have been only approved for hematological tumors because there seems to be a problem with the infiltration of CARs in real tumors, and that's very well represented here in our panel of patient-derived xenografts. To overcome this hurdle, I cannot disclose how we have armed these CAR Ts because we are currently protecting the intellectual property associated with this assay. What we have done is to make these CAR Ts more efficacious by arming the T cells. Basically, we make a more sophisticated construct in which we constitutively express the CAR, the tumor-specific CAR, in this case P95HER2, and we in the second example, we introduce an inducible promoter that is only turned on when the CAR is activated, and that increases or triggers the expression of something else that has anti-tumor or that increases the anti-tumor efficacy of these CARs. This is what, if you can read any reviews, this is called like tracks. And we are very happy to show that these tracks that we are making are very, very efficacious against patient-derived xenografts. This is an ongoing experiment comparing the CAR Ts. Again, in red, we have some anti-tumor effect, but when the CAR is armed, then the tumor effect increases very much. And we are following up to try and see if this response is so durable as the response that we have seen when analyzing the growth of cells expressing P95 cell HER2 of cell lines. And this takes me to the last part of the presentation. As Adriano has mentioned, we are also very interested in developing, these CAR Ts against P95HER2 will be tested in a clinical trial starting in a couple of years. What we are also very interested in parallel is into developing models of resistance against redirected lymphocytes. And no matter if we redirect lymphocytes with T-cell bispecific antibodies or CARs, we are sure that we are going to have models of resistance. One of the models of resistance that we are not interested in is the down-modulation of the antigen. That's very straightforward, and we have seen that we have observed it in cell lines. But HER2, the tyrosine kinase receptor, that is a very potent driver, is a very nice system to unveil other mechanisms of resistance. Since it is a driver, it cannot be down-modulated. So that's not a mechanism of resistance that we expect on cells expressing HER2. So that is what we did. We took cells expressing HER2, and using the PBMC model, we treat them with the T-cell bispecific antibody. And after treatment, we allow the tumor to regrow, and we repeated this treatment 3 times. After 3 times, we isolated tumors that were completely resistant to T-cell bispecific antibodies against HER2 or against HER2 CARs. I will not show all the data, but the cells are resistant to both CARs and TCBs. As expected, as you can see in the blue and yellow bars, there's no down modulation of HER2. As I have already mentioned, HER2 is a driver, so the cell very likely cannot down modulate it if it has to grow. And I will not go into the details, but we were able to show that the down modulation of JAK2 was the cause of this resistance. As you know, JAK2 is one of the molecules that transduces the signal of interferon gamma, and it turned out to be very important for the killing mediated by TCBs and CARs. Again, trying to expand on these results, we did something similar with one of the PDXs that is HER2 positive and grows into mice humanized with hematopoietic CD34 positive cells. We were able to humanize these mice, as I have already shown. After several weeks, they have plenty of human T cells, and the mice do not develop this graft versus host. So we are able to implant, in this case, a PDX, and then treat the mice with TCBs, allow the tumor to regress, allow it to grow again, and treat them again. Again, after 3 or 4 rounds of treatment and allowing the tumor to regrow, we were able again to isolate cells that were completely resistant to TCBs or to CARs. Again, as expected, and shown with the blue and red bars, the modulation of HER2 was not an option for this tumor to continue growing, and we observed the same downmodulation of JAK2. As I said, for the sake of time, I will not go into the details, but basically, this is the mechanism of resistance that we described in these models of resistance to HER2. In normal cells, the secretion of interferon gamma is very important for the T cells to kill the target cells. In a normal cell, JAK2 is an important mediator of this cell death. In resistant cells, JAK2 is downmodulated, very likely epigenetically, and the downmodulation of JAK2 turns off interferon gamma signaling, and there is enough for the cells to resist the killing of redirected lymphocytes, redirected via TCBs or redirected via CARs. If you want to expand on the mechanism, I would be very happy, but since the focus of this talk was the use of mice to test this resistance, I will not go into the details. So finally, I would like to acknowledge the members of my lab that all this work, Irene Rius and Macarena, they co-developed the T cell bispecific antibodies and the CAR Ts, and Enrique and Alex, I cannot point at them, but anyway, Enrique and Alex were able to develop all the mechanisms of resistance and show that the modulation of JAK2 is a quite frequent mechanism of acquisition of resistance against redirected lymphocytes. And Christian Klein from Roche is our long-term collaborator. We have co-developed the T cell bispecific antibodies, and we are still collaborating. And last but not least, as I have shown you, my lab is in 2 places in Barcelona, and the place by the seashore at Hospital del Mar. By the way, mar means sea in Spanish, so that's why it's Hospital del Mar. Has this institute of research that is in a very nice location, and we have positions available for postdocs and pre-docs. If you are interested, just please send me an email to that address, and we can discuss on future possibilities. So thank you very much.